In this video, we will discuss an important essay questions on enzyme inhibition. So the question is explain different types of enzyme inhibition with suitable examples. So first of all, just enzyme inhibition is of two types: reversible, irreversible, and different types of reversible and irreversible inhibition. And just write a few points regarding reversible inhibition and irreversible inhibition. And the major type of inhibitions are: one is competitive inhibition. Here you should write what are the features of competitive inhibition. In competitive inhibition, the inhibitory molecule is structurally similar to the substrate, so they bind to the active site, thereby inhibit the binding of the substrate. So once they interfere the active site, that will affect the Km value. So here the Km value increases. And this, after writing all these points, just draw the enzyme kinetic curve. So the first graph is double reciprocal plot, and the second graph is enzyme kinetic curve. So here the x-axis represents the substrate concentration and y-axis represents the reaction velocity. Here the important thing is Km value increases in competitive type of enzyme inhibition, whereas Vmax remains same. There is no change in maximum reaction velocity attained by the enzyme molecule. After that, you should write at least two examples. Here the best example is the drug which is used in hypercholesterolemia, that is the atorvastatin. Here the atorvastatin group of drug inhibit the HMG-CoA reductase enzyme of the cholesterol biosynthesis. And the second type of drug is methotrexate. It's an anti-cancer drug, and that methotrexate acts as a competitive inhibitor of folate reductase enzyme. Then just write the second type of inhibition, that is non-competitive inhibition. Here you should write these points, that is this inhibitory molecule is not structurally similar to this substrate, so they have no affinity towards the active site, and it is usually a irreversible type of enzyme inhibition. And what are the enzyme kinetic curve in non-competitive type of enzyme inhibition? Here the Vmax decreases, but Km value remains same because the active site is not interfered by the inhibitory molecule, so there won't be any change in the affinity of this enzyme molecule towards its substrate. So Km value is not changing non-competitive type of inhibition, whereas Vmax decreases. After drawing this graph, just write two examples. Either write the example as cyanide, that inhibiting the cytochrome oxidase enzyme, or fluoride, which inhibit the enolase enzyme of the glycolysis. And the third type of inhibition is suicidal inhibition. In suicidal inhibition, the inhibitor initially it is a part of this reaction, and after undergoing few reaction, that inhibitory molecule is converted into the active component. Once it is activated, that inhibitory molecule promote the degradation of the enzyme. So it is a type of irreversible inhibition. The best example is the allopurinol, that is a drug of choice in hyperuricemia or gout, where the allopurinol inhibit the xanthine oxidase enzyme. And the next type of inhibition is uncompetitive inhibition. The important thing is that inhibitor has no affinity towards the free enzyme molecule. The inhibitor binds to the enzyme substrate complex, and in this case both Vmax and Km value decreases. This is the enzyme kinetic curve in case of a uncompetitive inhibition. The important thing is the x-axis represents the substrate concentration and y-axis represents the reaction velocity. It's a double reciprocal plot. So the x-axis represents the reciprocal of substrate concentration, and y-axis represents the reciprocal of reaction velocity. And another type of inhibition is allosteric inhibition. It is a type of regulation, allosteric regulation. If the regulatory molecule inhibit the enzyme activity, then it is known as allosteric inhibition. Here the inhibitory molecule binds to the allosteric site of enzyme molecule. Here the enzyme molecule having both active site and the allosteric site. This allosteric molecule binds to the allosteric site of this enzyme molecule. Both are different, allosteric site and the active site. Substrates bind to the active site, whereas the allosteric regulator binds to the allosteric site. Here the enzyme kinetic curve will be a sigmoid in shape. And just write the points related to the allosteric inhibition. And the last type of inhibition is feedback inhibition. Here the product itself inhibit the corresponding enzyme. Here the example is glucose 6-phosphate. That product inhibit their formation, that is the exokinase enzyme. Exokinase enzyme mediate the formation of glucose 6-phosphate. If there is excess amount of glucose 6-phosphate, that product will inhibit that enzyme molecule. That type of inhibition is known as feedback inhibition. So that's all regarding the enzyme inhibition. Just write the features of this type of inhibition and draw the enzyme kinetic curve, and just write at least two examples. So that's all regarding enzyme inhibition. Thank you.